Okay, let's get started. We're aiming to finish by just after 1, to give people enough time for lunch and to get away on time this afternoon. So welcome to the last session. We'd like to invite the first speaker to present, Thomas Joris from University of Liège on BLV.
Thank you for the introduction. First of all, I don't have any conflict of interest in regard to this presentation. So I'm interested in BLV, which is a retrovirus that naturally infects bovines, but also share a lot of similarities with HTLV1. As you can see, the genomic structure with the provirus is fairly similar, and also, and importantly, the long terminal repeat, or LTR, are both able to promote a sense and antisense transcription. For the bovine leukemia virus, antisense transcription is promoted by the 3' LTR and generates 3 different antisense RNAs, ASS, AS1S, AS1L, and AS2. The sense transcripts are exported outside of the nucleus, where they can be either translated into the new proteins or incorporated into newly forming variants. For the antisense transcription, however, they are mostly retained in the nucleus, and at the moment, we still don't know what their biological function is. So that's what is the aim of this research, is to understand a little bit more about antisense transcription of the bovine leukemia virus. Our approach was based on a reverse genetic approach, and so we needed a valid target to only affect antisense transcription. Luckily, we exploited a mechanistical difference during the initiation of sense and antisense transcription. The initiation of sense transcription starts with the binding of the tata binding protein, or TBP, to the tata box that is located in the U3 region of the 5' LTR. For antisense transcription, the mechanism is not fully understood, but we know that antisense transcript initiates from the R region as well as the U5 region of the 3' LTR. Other labs have found that there are actually TF2B recognition element, BRE, as well as a motif 10 element and downstream promoter element, DPE, located in those regions. So these were valid targets for our reverse genetic approach. We tested different mutations. I'm only going to talk about the A1 mutation here, but we tested them with those kind of construct, which is simply a plasmid encoding different luciferase gene in different sense of transcription. So Renina for antisense transcription and Firefly for sense transcription. Our data showed that the A1 mutation was able to significantly decrease the antisense transcription activities, and this was the case in absence, but also in presence of the protein tags. Interestingly enough, the sense transcriptional activities, as measured with the Firefly luciferase, also showed that there was a significant increase in sense transcription. So then we wanted to see if the A1 mutation affected viral infectivity, and so we constructed a complete provirus with that A1 mutation located in the LTR, and we measured the impact on viral infectivity using 2 different co-culture experiments. So we transfected egg cells with either the wild type or the A1 provirus, and then we co-cultured them with reporter cells OVK-U3-LUC, which are cells that contain a Firefly gene under the regulation of the U3 region of the BLV-LTR. So this is inductible by TAX protein. And in that experiment, we saw that there was a modest but significant increase in the luminescence observed in the co-culture with the cells transfected with the A1 mutant provirus. The second co-culture experiment that we did was simply a syncytia formation assay, and in that experiment, there wasn't any difference between the 2 co-cultures. Then we inoculated 2 different groups of sheep, one with the wild-type provirus and one with the mutant provirus. And we saw that the A1 mutation did not change the infectivity of the virus. All the animals were infected by the virus and started to develop antibodies directed against the virus. But surprisingly enough, we observed that there was a drastic reduction in the viral replication as measured by the provider loads, as you can see. And also, and very importantly, there was no sheep injected with the mutant provirus developed any kind of lymphoma or leukemia, which was the case for most of the animals injected with the wild-type provirus. We then took a look at the lymphoid organs, and we measured the proliferation by immunohistochemistry labeling of the Ki67 proliferation marker. And in the lymph node, we saw that there was actually a decrease in cellular proliferation happening in the lymph node. And then we went to look at the spleen of those animals, and we saw that it was the opposite, which, yeah. So that was the opposite, with a significant increase in the cellular proliferation occurring in the spleen of the animals injected with the mutant provirus. So then, in order to understand a little bit more about how those phenotypes can be observed, we performed an RNA sequencing on the RNA extracted from the B cells, which is the cell targeting that BLV infects. And we saw that there was different genes that were up and down regulated, but there was one gene in particular that was highly upregulated when we compared the A1 injected sheep compared to the wild type, but also compared to non-infected control. So NI means non-infected. And that gene in particular was HMCN1, as you can see here and here. We also confirmed that observation with a separate RT-qPCR experiments on PBMCs collected from those animals. So HMCN1 or hemisentin-1 is a protein that is present in the extracellular matrix, and it actually plays a role in tissue structure as well as adhesion between two different types of tissues. Importantly, it is present in the lumen of lymphoid conduits, and another role that can be associated to hemisentin-1 is a role in cell division. So it actually promotes the division of the two daughter cells at the end of mitosis. We looked by confocal microscopy to the PBMCs, and we saw that there was an increase of the protein HMCN1 inside the PBMCs in the PBMCs collected from the sheep infected with the mutant compared to the wild type. And we also observed more protein from immunohistochemistry staining from the spleen samples. So higher, as you can see, there is higher amounts of hemisentin protein present there. Finally, we wanted to understand a little bit more about the significance of having HMCN1 gene dysregulation in the HMCN1 gene expression. And so we used RNA interference in different cell lines. So we used two shRNAs to knock down the HMC1 gene expression, and then we cultured those cells and we measured the migration of those cells. And as you can see, in both cell lines where the HMC1 gene was knocked down by RNA interference, there was actually a decrease in cellular migration. So in conclusion, our results show that we have a mutation that selectively decreases antisense transcription in cell culture that does not impact cell fusion, but slightly increase infectivity. And then when we injected sheep with that mutant, we saw that there was an increase in cellular proliferation in the spleen, as well as a diminution of viral replication inside those animals. That phenotype was also correlated with a increased expression of the hemisentin-1 gene, and importantly, none of the sheep developed any kind of leukemia or lymphoma. So I would like to thank my lab for this project. I would like also to thank the organizing committee for choosing this abstract, and I would like to thank you for your attention.
Thank you very much. And the next speaker is Morel Sotomi from Imperial College London. His title is Characterizing elite controllers. Please.
Okay, so thank you. My name is Morel. I'm a current master's student here at Imperial, but today I'm going to be talking to you about work I did in my undergrad under Carolyn Graham, looking at characterizing HTLV1 elite controllers using a range of molecular techniques. So firstly, when we think about this concept of elite controllers, this is something that is well defined for HIV, and there can be some differing definitions in the literature, but it's generally described as patients who are able to spontaneously maintain an undetectable viral load in the absence of antiretroviral therapies, and this is usually established soon after seroconversion. And this is also distinct from patients who are long-term non-progressors. However, for HTLV1, there's currently not an elite controller definition. However, there would be some key differences. So firstly, it would be very low / undetectable proviral load rather than viral load. And currently, antiretrovirals are not used in the clinical routine for HTLV1. However, it's also important that these patients are also seroconverted for HTLV1. So to try and identify a population of these patients, we came up with a preliminary elite controller definition. So firstly, these patients had to be positive with an immunological assay, so Western blot or line immunoassay, a proviral load that never crossed 0.1%, a minimum 2 years of follow-up, and also minimum 2 time points where they were both qPCR and nested PCR negative. Now, using this definition, we're able to identify 9 people living with HTLV1 as potential elite controllers, and importantly, all of these patients were Western blot positive, so they were confirmed to have been infected with the virus as they have antibodies against viral proteins. However, their PCR results were very low, undetectable, suggesting they had low levels of the virus in the blood. And this was occurring below the detection limit for both qPCR and nested PCR. So some of the questions we had from this was, are there actually issues with detecting HTLV1 provirus in these patients, or can they actually effectively control the viral replication to the degree that we're not detecting it in their blood? So we hypothesized that this undetectable, such very low proviral load results were true results, therefore suggesting that these patients can actually effectively control viral replication. So just some key 2 points to remember before continuing. One is that a HTLV1 elite controller definition will differ from HIV, and 2 is that the patients identified as potential elite controllers were all confirmed to have been infected with the virus. So some of the ideas we potentially could have gone wrong is, do these patients have a mutated provirus? So do they have a mutation in the TAX primer binding site, and is this why we're not detecting them? And can we actually increase the detection sensitivity using different molecular techniques? So in regards to the mutated provirus, we use alternative TAX primers, which you can see here are targeting different regions of the HTLV1 provirus. So if these patients had a mutation in the routine TAX primer binding site, we may be able to detect them using these alternative TAX sets. And in regard to increased sensitivity, we used digital droplet PCR, which is reported to potentially have increased sensitivity compared to qPCR, and also CD4 positive purification, as it is known that HTLV1 primarily infects CD4 positive lymphocytes. So to validate these assays that I've mentioned, we use samples from 25 people living with HTLV1 with a range of proviral loads and disease statuses. And once we had validated the assays, we then moved on to looking at these potential elite controller patients. So first, if you look at the alternative TAC primers, we took the whole blood from these patients, separated out the PBMCs, extracted the DNA, and then retested using the routine TAC set, as well as additionally with these 2 alternative TAC primers. And if we look at the results here, we can see that there is not a significant difference in the proviral load obtained using the 2 alternative TAC primers when comparing this to the routine TAC set. So we're able to consistently detect HTLV1 provirus across different TAC primers. And supporting this, we see a strong and significant correlation in the proviral loads obtained between the 2 alternative TAC sets comparing to the routine TAC set. Now, if we look at the digital droplet PCR, this was done in the same way as the qPCR, with kind of the only notable difference really being in the housekeeping gene used. Now, if we then add the digital droplet PCR results, we can see that we're getting a significantly higher proviral load obtained using digital droplet PCR compared to both the routine tax set and also to these 2 additional tax sets I've mentioned. Now, despite this higher proviral load obtained, there is still a strong and significant correlation between the proviral loads obtained using digital droplet PCR as well as qPCR. Kind of again suggesting that we're able to consistently detect HTLV provirus across different molecular tests. Now moving on to the CD4 positive purification. We took the PBMCs stored in the tissue bank for these patients, CD4 positively magnetically sorted them. We checked the CD4 positive purity using flow cytometry, and then extracted the DNA from this CD4 positive enriched population, and then retested using the routine tax set with qPCR. Now, if we compare the proviral loads obtained using PBMCs and CD4 positive cells, we see that we're getting an expected significant increase in the proviral load obtained in the CD4 positive enriched samples compared to PBMCs. And I'm saying expected, as I mentioned earlier, HTLV1 primarily infects CD4 positive cells, so we expect that by enriching this population, we'll be able to increase the proviral load. So now that I've shown you how we validated the assays, now let's look at what happened when we tested the potentially controller patients. So here I'm showing you a table of the number of positive replicates obtained using PBMC, when testing PBMCs and CD4 positive cells. And these patient samples were tested up to 6 times. And kind of what I'm highlighting here is that the majority of these replicates for the potentially controller patients remained undetectable. And even when detected in the PBMCs, the proviral load was never greater than 0.03%. So we can say that these potential elite controller patients were consistently had low or undetectable proviral loads, giving us strong evidence that these patients could actually effectively control viral replication, and they were elite controllers. So some key take-home messages. We were able to consistently detect HTLV1 provirus across different molecular tests, and we were able to use this to identify a group of people living with HTLV1 that can actually effectively control viral replication. Now moving forward, it is also important to test other regions of the HTLV1 provirus. There's also potential epidemiological questions for these patients. So does the method of how they were infected with the virus, does this potentially change elite controller status? This is not the case for HIV, where you see a diverse range of transmission routes, but it could be the case for HTLV1. And it's also interesting to compare the immune response of these elite controllers to other people living with HTLV1, which could potentially better aid in treatment development. And I've highlighted vaccination here, because it is known for HIV elite controllers. They're actually targeting different epitopes of HIV, and this is potentially why they're able to better control the virus. So this could similarly be the case for HTLV1. I think it's also essential to assess the transmission risk of patients with low proviral loads. And currently, there is no proviral load threshold where below transmission is negligible for patients, and this is a particular concern for the WHO. It is only known that with increased proviral load comes increased transmission. However, I'll echo something here that has been mentioned throughout the conference, is that to properly assess the transmission risk for these patients, as well as to develop a universal elite controller definition, it's essential that we develop a commercial molecular assay for HTLV1. And now to provide a general summary, we took these 9 potential elite controller patients who were confirmed to have been infected, but had low detectable levels of the virus in the blood. And we thought, are there actually issues with detecting these patients in the blood, or can they actually effectively control viral replication? Is this why we're seeing these low results? And after validating multiple different molecular assays, these patients remained very low or undetectable, suggesting that they could effectively control the virus and were elite controllers. So thank you first and foremost to the people living with HTLV1 for donating their samples, but also F1 at the Imperial College NHS Trust, as well as F1 at the Department of Infectious Diseases. Thank you.
Okay, so next speaker is Professor Lee Ratner. So the title is The Role of CTCF Binding Site in the HTLV1 Pathogenesis in Vivo.
Okay, I'd like to thank the organizers for giving me the opportunity to tell you about our work on the role of the CTCF binding site in HTLV pathogenesis. And I have no conflicts related to this presentation. So as you've heard, CTCF is the best characterized insulator binding protein in eukaryotes. It plays an important role in the 3D structure of chromatin, as well as playing a role in transcriptional activation, repression, or silencing. CTCF interacts with or regulates several DNA viruses, including HSV, EBV, KSHV, CMV, and adenovirus. In HTLV1, the virus integrates in the host cell and includes a single CTCF binding site based on the work first reported by the Bangham group. So we established infected cell lines using HTLV1 lacking the CTCF binding site and assessed the impact of inhibiting the interaction of CTCF with the viral genome. To do this, we mutated the consensus CTCF binding site in a mutant HTLV1 delta CTCF, as shown here in red. But these mutations required also an alteration of the P12 coding sequence. So to correct for that, we actually truncated P12, eliminating the last 27 codons of the P12 open reading frame, and that portion of that protein is not known to have any significant biological functions. And in fact, the STLV1 P12 is truncated at a similar position. To make a comparable control wild-type clone, we made another P12 stop clone that still had the CTCF binding site, but had the stop codon for P12. So in our previous work published in PLS Pathogen, we deleted the CTCF binding site and found that this resulted in an expansion of DNA methylation into the PX region, as well as differentially increased trimethylation of H3K4, K36, and K27, actually throughout the provirus. By inhibiting CTCF binding to the HTLV1 provirus, we could see that it actually affected proviral gene transcription, and in 2/3 of the single clones carrying a single infectious virus, we could see that shRNA to CTCF increased TAX expression. In 1/3 of the clones, there was no change with reactivation of HTLV expression. In no cases did we see lower TAX expression when we knocked down CTCF binding. So to investigate the role of the CTCF binding site in vivo, we used humanized mice. And for today's presentation, I'll show you work with CD34 hematopoietic stem cells based on the Duke-Dudon model, where they're injected into the liver at 0 to 2 days, then allowed to humanize for 13 to 15 weeks, infected with irradiated HTLV producer cells, and then followed for an additional 13 to 16 weeks. We have very similar results with CD133 positive hematopoietic stem cells injected in the tibia at 4 to 5 weeks of age, and then infecting the mice at about 20 weeks of age, a model developed by Fujisawa and colleagues. And we monitored blood every 2.5 weeks for proviral load and monitored the mice for ATL-like symptoms. So as you can see, with mice infected with the CTCF mutant, out to 12 weeks, only about 30% of the mice developed disease, and they developed disease at a much slower rate than either mice infected with the P12 stop mutant or the wild-type clone, all of which developed disease by 4 to 5 weeks of age. You can see that the CTCF mutant infected mice had a lower spleen weight, lower absolute lymphocyte count, and at the time of necropsy, although the levels of CD4 infiltration in the blood were very similar, I just want to point out that necropsy was done at a much later time point in the CTCF mutant knockout infected mice compared to the P12 and wild-type infected mice. We saw lower levels at necropsy of CD4 infiltration in the spleen, liver, and bone marrow in the CTCF mutant infected mice. Measuring proviral load at 2.5 weeks, we can see that very, very low or undetectable proviral load was seen in all of the CTCF mutant infected mice compared to the large majority of the P12 and wild-type infected mice. And the proviral load at 5 weeks overall was quite low still in the CTCF knockout infected mice. And the proviral load in the blood, spleen, liver, and bone marrow was consistently lower at the time of necropsy in CTCF mutant infected mice compared to P12 stop mice. When lymphoid disease developed in wild-type P12 stop and CTCF mice in the spleen and liver, you can see diffuse infiltration of lymphocytes, which were CD4 positive, as seen by immunohistochemistry. And this is compared to the same CD4 IHC performed on normal tonsil and 2 different ATL patient samples. We subdivided the CTCF mutant mice into the groups that had absolute lymphocyte counts of over 400 and those with normal absolute lymphocyte counts. And in the first group of mice, these were the only ones that developed disease, again, very late and only in a small proportion, 30 to 40% of the mice. And the mice that developed disease had splenomegaly, higher proviral loads at 2.5 and 5 weeks, higher levels of CD4 infiltration in the blood, spleen, liver, and bone marrow at the time of necropsy, and the proviral load in the PBMCs inversely correlated with the survival of the mice. We performed single-cell RNA-seq analysis on splenocytes obtained from these humanized infected mice. And we could separate the human cells from the mouse cells and identify human T cells that were also CD4 positive, CD8 positive, CD25 positive, or expressing TCR. We saw a small number of cells that were expressing detectable HTLV transcripts, and the only HTLV transcript we could detect were HBZ. We could not detect any plus-strand transcripts in this model. In the CD4 lymphocytes, we could see overexpression of genes associated with ATL in the CD4 cells, including CADM1, FOXP3, CD28, CTLA4, and IL-2 receptor alpha, and a number of other genes as noted here by string analysis. When we compared the results from splenocytes from 3 CTCF mutant-infected splenocytes to those from P12 stop-infected splenocytes, you can see there were higher levels of CD8 cells in particular, and CD8 cells that express granzyme B, higher levels of T cell activation markers, PTRCAP and IFIT-M1, as well as markers of cytotoxic vesicle release, including granzyme B, NKG7, and GDNLY. We saw higher levels of ALOX5AP, which is associated with inflammation, expressed on T cells in the P12 infected splenocytes compared to the CTCF infected mouse splenocytes, but lower levels of TCR genes that are expressing a CDR3 sequence that match that in a CTL epitope that is known to be reactive with TAX. To try to understand what's going on, we also looked in vitro at early steps of infection. Here, infecting jet cells, as you heard, carrying RFP, and then sorting those cells. So at 72 hours, you can see lower numbers of RFP positive cells compared to the P12 stop or ACH wild type infected cells. And by incucyte analysis out to 5 days, you can see higher levels of red objects, RFP per well, in the P12 stop cultures compared to the CTCF infected cultures, at least over the first 3 days, and then similar levels per well after that. If one then looks at the RFP level per cell within the wells, you can see higher levels in the P12 stop compared to the CTCF mutant cells, infected cells, out to about 2.5 to 3 days, and then lower levels subsequently, whereas in the CTCF infected cultures, just a slow, steady increase over time. We think this may be similar to what is occurring in vivo and explaining the in vivo results. So our working hypothesis is that when the CTCF site is present, you have early infection, which is rapid onset acute disease characterized by elevated tax production, virus production, lymphocyte expansion, antigen presentation, and cytotoxicity, and then subsequently perhaps more suppressed expression. Whereas when there's loss of CTCF binding, there's a more smoldering infection with low levels of all of the parameters that I just mentioned, or no disease whatsoever. And whether this correlates to what appears in humans with smoldering or asymptomatic individuals compared to rapid and acute disease remains to be investigated. So in summary, mutation of the viral CTCF binding site delayed virus spread and abrogated or delayed lymphoproliferative disease. Multi-omic TCR evaluation showed clonal T-cell expansion. Single-cell RNA-seq showed gene expression patterns in CD4 positive T cells similar to those in ATL. CD4 and CD8 T cells, particularly CD8 T cells in vivo, were enriched in T cell activation factors. And temporal expression of TAX demonstrates dynamic regulation of the viral CTCF binding site, perhaps consistent with the in vivo results. And future studies hopefully will uncover the mechanisms underlying this CTCF binding site interaction with TAX expression. So just a lay summary that these infected humanized mice develop an ATL-like picture. Pathogenesis is regulated by this insulator protein called CTCF. Within hours after infection, the binding site regulates the viral oncogene expression for TAX, and then hopefully we'll understand more about the mechanisms in the future. So I'd like to particularly thank Ansi Joseph, Shogun Chang, John Harding, and Dan Rao in my group, as well as Deb Weiss, particularly for the pathologic and bone analyses, and then Pat Green and Jacob Al-Salim at The Ohio State University Center for Retrovirus Research. Thank you.
Can we get the speakers to come to the front, please? While you're doing that, just to start off one of the discussions, we still don't fully understand the role of CTCF binding to HTLV1. I think Lee's report is very exciting. I'd like to suggest we might be able to bring it together with one of the other talks, which was by Akhil Rahman. That is, it may be that the insulator function of CTCF is blocking the ability of the silencer region to silence the 3' LTR, and so maintain almost constitutive expression of the minus strand, but to maintain for most of the time silence of the plus strand. That's a suggestion. Lee, do you want to respond? Or?
Thank you.
And one question that arises is, how does the 5' LTR then overcome the silencer region? Is it by a positive disengagement of transcription factors, or simply does it overpower the silencer region? What is your current theory?
So far, we don't have data to comment on that, but we want to look into it.
Yeah.
Thank you.
Lee.
Yeah, so I think that's very interesting. The one thing we were wondering is the early effects that we're seeing, whether they're pre or post-integration.
Really?
And we were particularly interested in the work of Steve Goff that showed binding of the Hush complex, and whether CTCF actually could regulate that is another possibility. Yeah. So doing the IncuCyte analysis in the presence of integration inhibitors would be very informative.
Right. Just one other thing briefly on CTCF. As Lee's group has previously shown, the effects in a given clone can differ quite a lot from another clone. And of course, that is likely to be because it will pair with CTCF bound to the flanking host genome, which is different for every integration site. So it'll be very heterogeneous between clones. That's enough for me. Yeah.
Okay, this question's for Morel. Do you know what the clonality looks like in your elite controllers compared to ACs?
Yeah, that's a good question. That's not something we've looked at yet, but I think that's a good point. I think potential limitations is that we have kind of few number of cells from these samples, so it's hard to do all the experiments you want to do, but yeah, that's an interesting suggestion.
I mean, do you have enough material to be able to do that analysis? I mean, since it's so low?
It's too low. It'd be too low.
It's too low.
Yeah. I'm sorry, I don't want to keep on too much, but a brief comment on the elite controllers. We, obviously one possibility, as you said, is the efficient immune response. In the work that we did with Dr. Asame many years ago in Kagoshima, we found that there was a frequency of people in which the proviral load was below the detection limit. We could not look at their CTL response because the CTL response is too low. However, the immunogenetics showed a strong positive association between HLA-A2 and the occurrence of the detection limit, below the detection limit. So it strongly suggests that you're right, it's the immune response. Sorry.
Next question. Please.
Hello, I had a question for Amanda. So my name's Tiana, and we develop siRNAs against the LTRs, TAX, and HBZ. So my question was about your off-target effect analysis. So that was a really interesting approach. We found that different cell types had different off-target effect profiles with our siRNAs. I noticed you did yours in HeLa cells. Would you anticipate a difference if you move that into lymphocytes? Or is there a plan to do that?
I mean, it's possible. I think we started with HeLa because that's the easiest model, obviously. And then, we can go into KIT225 cells, IL-2 dependent T cell line at least. But yeah, I mean, maybe. Not sure until we do it. Yeah, thanks.
Okay, please. Go ahead.
I'm Jay from the University of Queensland. I had a question for Morel. I thought that was a really stunning talk. Sort of related to the other question that was asked, do you know anything immunologically about the people who, what do they call them, exceptionally well responders or something like that? Or even the somatic genetic information about those people?
Can you repeat the question, please? No, well, either you, yes.
Sorry, can you repeat the question?
Do you have any information about the immunology of these people, or the somatic genetic differences with normal?
So we don't currently. I think this is kind of quite preliminary work just to kind of show that these patients actually do exist. But I think that is an interesting suggestion to look at. Yeah.
Any other questions? So we received so many questions from the web, and one question is Lee. Have you quantified the HBZ tax messenger RNA or protein expression in the CTCF minus mice?
In the mice, we have only we only did single-cell RNA-seq thus far. We have no protein studies and no bulk RNA-seq. So, it's relatively insensitive, but thus far we only see HBZ RNA. And the number of cells are very small, so we really can't compare from the single cell. We'll have to look at bulk RNA-seq. And again, we don't see tax RNA or protein by tax RNA by single cell.
I see.
It would be great to be able to repeat your kind of experiments in an immunocompetent animal. As a challenge for you.
I mean, we've looked in rabbits with the CTCF deleted virus.
With these contrasts?
Yes.
Right, okay.
Yeah,
okay.
and we unfortunately didn't see a difference in persistence.
You don't?
We don't, not with proviral load, but if I remember the study right, Pat, I think we did see a difference in total antibody response, but not to the virus, to just total IgG levels were changed.
No difference in proviral load?
No. I know, we were surprised as well. That was not what we expected. We didn't look at clonality, though, or...
Still prefer primates to rabbits, but I know you like.
So, well, great presentation for all speakers. This is for Dr. Basarbachi. You know, in the management of ATL, working with Dr. Ramos in Miami, and your center, interferon has, we believe, a very good role. I mean, we see it in our patients, especially those that are no bulky, primarily leukemia. So your work is great because it kind of tried to narrow the gap about what we thought initially on what's the role of tags after the integration of the virus and then what happened afterwards. So my question comes into, do you have, or you're thinking, which we may have some samples too, and looking on maybe pre-pulse interferon, see what exactly happens there, for example, with tags or HBZ, because it may be some travel-driven things, especially for patients that are maybe no, will be a good question for me clinically, that are not maybe transplant eligible, in which maintenance therapy is going to be needed, and perhaps if there is any role there, or for example, Dr. Ratner trial too with lenalidomide. So what do you think would fit into there if you're able to continue demonstrating that actually activating the virus somehow, or even having some antigenic responses to the disease to control it may play a role, especially in patients that may need some maintenance therapy, those that are not trial or transplant eligible, for example.
So thank you. I think these are two important questions, but the target is not the same. When we use AZT interferon, we are inhibiting viral replication, which through RT, which is not happening in the leukemic cell themselves. I think that HTLV1 virus, but this is only a hypothesis and not demonstrated, is a high-level retrovirus reproducing at the cellular level what low-level retroviruses are doing. So oncogenic retroviruses are defective for replication, and they cannot survive without the help of a helper retrovirus. And I think that in ATL patients, the defective viruses require the de novo infection of T cells, and because ATL never occurs outside the setting of HTLV1 infection, and the newly infected T cells will produce survival cytokines and other factors to maintain the clone alive, and this is what you block with AZT interferon. So going back to targeting tags, unfortunately, the only treatment that has been shown to target tags so far was arsenic interferon that we developed, which works as to eradicate minimal residual disease, but no other therapies were developed. And I think that one reason was that people did not believe that tags is needed to maintain ATL, and this has delayed the development of tags-targeted therapies.
Okay, thanks very much. Got a couple more questions briefly. Do you want to?
Quick question for Lee. So your P12 stop virus seemed to outperform, as a the control outperform the wild type. Can you exclude that there's no impact on HBZ with that construct?
Yeah, not at this point. I mean, we did resequence all the virus that came out, and there were no reversions or changes. We didn't actually resequence the whole genome. And again, we don't have enough depth of HBZ sequences to be able to see whether there's any changes, but looks like the same HBZ that we put in based on the single-cell RNA-seq data thus far.
This is a question for Amanda, based on Mr. Aman's work. Did you score in your CRISPR-Cas9 screen any guides in that silencer region that they had, and would that have had a phenotype in your assay?
Well, let me ask her a question. Does the silencer region encompass any of the coding regions of HBZ or TAX? Because that's all we focused on was the coding regions of TAX and HBZ. So There, it obviously would have included the CTCF site, the enhancer sequence, but I'm not sure about.
It's from HTLV 4,300 to 4,600. So
I don't think
yeah.
that would. I'm thinking of the map in my head. I'm not thinking it would.
Okay.
So one question from web. So Ali, did you check provider status of your ATL cases? Did any of them carry the deletion of 5' LTR? And as has been previously described, and my question is also, how about the nonsense mutation of tags? In your case.
Let's try.
I heard part of the question. Is it about if we checked for the 5' LTR deletion?
Yeah.
So unfortunately, no. We got the cells from the Japanese ATL registry, and we performed the experiments on these cells. So we guess that when you have 56 ATL patients, 1/3 or so will have a deleted LTR, but we cannot tell which one. And actually, we're looking after more cells with a known LTR deleted or not deleted to increase the cohort.
Thanks.
I have one question to Mansky. So the future of immature the virus particle is similar. That happens in the other delta retroviruses, bovine leucan viruses, HTLV2 or simian T-cell leucan virus. So I guess that's probably a common feature of a delta retrovirus.
So are you making a comment or asking a question? I can't tell.
Are the other delta retroviruses the same?
Is what's the same?
You are immature viral particle? Yeah.
Well, we've looked at the immature particle morphologies at lower resolution for STLV and with BLV. We don't see any major differences, but we haven't looked at high resolution at this time. But it's a good question to ask, though. So.
No more questions?
Okay, I think we are drawing this session to a close. Thank you very much.